Okay, so continuing, so I want to focus on spatial evolution and the importance of spatial structure. And I will going to show some slides arguing that space is very important. I'm going to go through some papers, kind of zigzagging between other people's work, my work, since this is a tutorial, just to sort of show you why space could be important and studies that argue in favor of this. Space is important for a variety of reasons. If you look at solid tumors grow in terms of three-dimensional blobs of cells that have very strong spatial structure. So if you apply an ODE to model something like this, it's unclear what it actually does. If you look at in vitro growth models, organoids are becoming increasingly popular as an alternative to animal models. These are basically cell cultures where the tissue is taken from patients that maintain stem cells, more differentiated cells, even parts of the microenvironment, sometimes immune responses, and so on. But these are, again, 3D spatially structured populations that grow, which you can study experimentally. People often say that leukemias are liquid tumors, and therefore you can model them under the assumption of perfect mixing. But I don't think that's true either. If you look at this picture here, these are lymph nodes in a patient who has chronic lymphocytic leukemia. You do measure the leukemia cells in the blood, but nothing actually grows in the blood. Where things are growing are in those lymph nodes, and these lymph nodes are blown up, completely packed with tumor cells. Again, this is not a mixed system. Within each lymph nodes, there may be some mobility or not, but this is also a different spatial structure here where you have different patches. In ecological terms, you can say demes, where there are tumor cells in here, tumor cells in here, tumor cells in here. They're spatially separated, but they're connected to each other through the bloodstream and can potentially migrate from one lymph node or the spleen out into the blood, going to another lymph node or spleen.
The numbers are clones, or
sorry?
What, the numbers, they indicate the same clone? I mean,
the numbers indicate some volume or something. It's a radiological study. Yeah, this is just radiological study. Yeah, but it's just to argue that every tumor has spatial structure, and spatial structure can either be a population of cells like this or spatially distributed in different patches across the body. So modeling spatial structure is important. So I just wanted to outline a few modeling approaches. I will not be talking about most of them, just for completeness. How do you model spatial structure? There are partial differential equations where you can have a spatially explicit model. Then you can have agent-based models. They're more like cellular automata or individual-based models where you track individual cells, both in time and in space, and there are certain rules that apply. You update those models. Cellular POTS models are kind of a variation of these where you assume certain shapes of the cell, biophysical forces that are going on. And there are further variations, such as lattice gas cellular automata, where you can have more than a cell in a given spot. And then you can have hybrid multiscale models, where you have both agent-based models plus other diffusible factors modeled by PDEs, and so on. So this is sort of generally some of the more prominent modeling methodologies that you use to study spatial dynamics. What I'm going to be focusing on most during this presentation is either agent-based models, where you have individual cells that have a given spatial location. You apply certain rules to them. They divide. They can die. When they can divide to the nearest neighbors only. And so it's a very spatially structured system. You can compare the properties of such a individual-based or agent-based model to those of a model that assumes mass action, where everything mixes perfectly well, which is essentially what an ODE assumes. And you can, again, compare those things to these demes or patches where cells grow in spatially separate patches from each other that are connected to each other by migration. And one thing that I'm going to mention as I go through this, there can be a similarity between the properties of a patch model and the properties of these types of agent-based models. So I want to start off by highlighting some studies that show the important role of space for tumor evolution. This is a number of papers that I find interesting in the literature. There are many more papers that I find interesting in the literature that I'm not going to talk about because I only have this amount of time. So I want to start by talking briefly about a paper by the lab of Rob Noble. And what they did is they showed in a nice way how the population structure can be very important for evolutionary dynamics and for the fate of mutants as a tumor progresses. And they compared various different structures. There's non-spatial, where you see some patterns, and then there is, I want to highlight the fourth first, this boundary growth, where you have in the two-dimensional, this is represented in a two-dimensional setting here rather than 3D, but only cells on the surface contribute to a mutant growth, so it grows out like a range expansion. Then there are things like gland fission in adenoma, where you have crypts, where the whole crypt can divide, and that contributes to tumor progression. And then you have glandular invasion, which is again a different structural process. And they showed that each of those has unique signatures in which evolutionary dynamics can proceed. So I thought this is a nice paper to go to if you're interested in that, to get a good overview of why spatial structure can be important to study evolution. This is another paper that I found interesting from the lab of Alison Fader, and they did phylogenetic analysis to try to figure out how these tumor growth, and they found evidence of this sort of growth occurring where cells grow primarily at the surface rather than in the middle of a tumor. And I think that question popped up in the previous talk. Is there any evidence that tumors grow in this way? Indeed, and there's a lot of methodology involved, but the upshot of this study was that there was a significantly faster rate of division at the edge of the tumors that they found compared to the center of the tumors. And that was done in the context of hepatocellular carcinoma. So this is a nice study that provides evidence that this mode of growth is important. And I focus on this because I'm going to talk about this mode of growth quite a bit. We do quite a lot of our studies in spatial systems that look like this expansion. And here's a study from Arne Troelson's lab that shows that if you have a spatial structure like this, the way the mutants grow in a colony like this, if you want to sample the tumor to get an idea of what the genetic heterogeneity is in the tumor, it becomes really important what the sampling strategy is, how many samples you have to take in order to get an accurate picture of that heterogeneity. If you just do it randomly, you get a wrong picture, basically. So that's an important point. This is another study showing how this sort of 2D range expansion can be very important. This is from Sandy Anderson's lab at Moffitt. They talk a lot about adaptive therapy. So here you have a tumor growing. Stop working again. You have a tumor growing where the gray is sensitive cells, and in the middle of it, they grow drug-resistant cells. And if you knock this tumor with a treatment, a chemotherapy, for example, that eliminates all of the sensitive cells, you spatially release and set free those resistant mutant cells, then it can grow and relapse really quickly. On the other hand, if you treat with a lesser dose, they argue that you maintain some of these sensitive cells around and that they can then, in a spatial way, compete with the resistant cells and suppress them, and this results in a much longer relapse time of the resistance strain. Again, spatial dynamics very important, especially those range expansions that I mentioned before. And when you talk about range expansions, one of the important paper is by the group of Oskar Halacek, who showed very elegantly, both experimentally and with computational models, how these mutant populations can evolve in a spatial setting. So you get these sectors emerging that expand and grow at the surface. And then you can ask, what is a given mutant burden when the expanding colony of cells hits a given size, a size n. And if you compare it in a spatially explicit setting like here, compared to a mixed system, they found that typically in a spatial system, at the same threshold size, you have significantly higher number of mutants, a higher mutant burden compared to a mixed system. And that is a very important result because it is very often interesting to estimate the mutant burden at a given size when you start treatment. If you want to know, for example, how many resistant mutants are already there, how long will it take to relapse, and so on. So this was a very nice study. We did some expansion of this study. Look, this is just kind of similar things showing here. This is the average number of mutants in a growing colony. These are neutral mutants in the spatial system, many more than in the mixed system. This is for advantageous mutants. It's the same here as well. The interesting thing that we found was you can describe the same dynamics with patches, with these deme models that I mentioned before. So instead of having an agent-based model that you have here, where cells explicitly move to the nearest neighbors, you can have your cells live in patches, and I don't know what these patches can correspond to, either the lymph nodes in the medical terms, but in ecology, there could be different ponds that are in a given habitat. And individuals can move from one patch to another. Either they can move only to nearest neighboring patches, or they can move to any of the patches in the system. And interestingly, we found that for both these patterns, you see the same result as in the spatially explicit model. That is, you have more mutants if you have more neutral mutants and more advantageous mutants in the patch model compared to a mixed system. So here's the ratio of the model that you find in the patch model compared to those you find in a mixed system, and it's always greater than 1. And interestingly, it doesn't always happen or doesn't only happen if you assume nearest neighbor, so spatial migration. It also happens when you assume non-spatial migration. So we think it's not really a spatial thing as such, but a population fragmentation effect that leads to this higher number of mutants at the threshold size. There's some other interesting results here that came out of this study that that it can be quite complicated. If you look at deleterious mutants, the result actually depends on where your disadvantage is in. If a cell has a disadvantage in the division rate, then you have many more mutants. You can have many more mutants in the spatial system, the red, compared to the non-spatial system. But if the disadvantage is in death, then it's the opposite result. You have fewer mutants in the spatial system compared to the non-spatial system. So it's a very complicated thing. It's a complex dynamics, and it is important to take all of this account if you want to work your way towards being able to predict how many mutants are going to be there at a given threshold size in a tumor, for example. This is a very simple model, doesn't take any of the tumor complexity into account, but obviously a tumor is spatially structured, so it's going to build on this kind of framework. So that's, yes?
Sorry, so the advantage would be that the deme model is easier to simulate because you have a few demes and not many demes.
Well, that is definitely an advantage. Not to simulate, to get some analytical insights in as well. Right? To simulate, they're both pretty horrible to simulate, especially large populations. But you can actually gain some analytical insights because within each deme, you can have population dynamics that is based on Gillespie simulations of ODE, for example, and so that can be then tackled more easily. But it's also biologically important, I think, because a lot of, for not only in medical settings, I mentioned lymph nodes, but in ecology, there are lots of patch systems, right? And that applies to these sort of things.
So you would argue that if you have a biological system, pay attention to these demes more, or
yes,
as well as
true spatial.
Interesting.
Yes.
Okay.
Yes. Uh-huh.
So it looks like you were able to do a lot of analysis in like a two-dimensional slice. Were you also mentioned that it's a simple model. Were you able to look at like the three-dimensional slices and how those would create like similar to like a 3D lens?
Yes, 3D has similar properties. A little weaker the effect because 3D is more mixed because you have more neighbors. And I'm going to come through to that in a minute. Yeah. Uh-huh.
Sorry, it's my fault if I missed it. So the conclusions, the sort of message in there about the mutants and spatial compared to mixed and so on in the two cases, that was based on simulations with some specific parameters for everything, right?
Right. Yeah, with a big parameter sweep. Yes.
I see. So on a huge parameters.
Yes, and also you can get insights into the scaling laws. I'm going to talk about this a little bit.
Okay.
Scaling laws of the
mutants. You're confident that these are...
Yes. Yeah. Okay. So this is comparing number of mutants in a spatially growing population at threshold size to a mixed system. So took that a little bit further, you can derive scaling laws for, this is just not one mutation, but n-hit mutants. So a cell can have 1 mutation, 2 mutations, 3 mutations. They can occur in different dimensions, and they can lead to different fitnesses. So here you have scaling laws. For example, this is exponential growth, where you have the wild type and the mutant. That's pretty well understood. If you have 2D spatial growth, you can see that the mutant grows differently compared to the mixed system, which is the reason for these results that I previously presented. So we expanded this, looking at n-hit mutants, 1, 2, and 3-hit mutants for the neutral case. You can derive it's a power law, the way it grows. You can derive the exponent of the power law. You can do it for mutants with different fitnesses. So here you can have an advantageous mutant where the mutant gains an advantage, and here you have 2-hit mutants, either they're all neutral, or you always assume that a mutant is advantageous with respect to the previous type. So here you get one advantageous type, the other one is then not changed, and here you get 3 types of advantages. And you can do it for n equals 3 mutations as well. So that's an expansion, an extension of this work that we did. And why is that important? Because tumors are very large cell populations. They reach up to 10 to the 12 to 10 to the 13 cells. If you want to simulate it, and if you want to predict what is the number of mutants at 10 to the 12 cells, and do this by computer simulations, you'll be there for a very long time before you get any results. So it's impractical, it's not feasible. But with these scaling laws, you can basically run your computer simulation for a feasible amount of time, and then apply your scaling law to extrapolate how many mutants you're going to have at much larger sizes. And just an example of how this can be done. Here is an application of this approach to an evolutionary question. And this evolutionary question arose through a collaboration we have with a guy called Justin Pritchard at Penn State. And he was interested in the effect of gene duplications on the rate of evolution. So what we're looking at is evolution in a given gene. Like, imagine a point mutation. The most straightforward way to get this point mutation is go from your wild type, introduce the mutation, and you get the mutant over here. That happens at a relatively slow mutation rate, because mutation rates are low. What can happen is you can have a cell with gene amplifications. Duplication is 2, doubling it. You can have more amplifications. If you've got more copies of the gene, you have more copies that you can mutate, so your mutation rate becomes higher. So going from here to the mutation that you're looking for is a faster event, okay? And it is faster because you've got more copies. So the question is, what is faster? What is more important, going directly from the original wild-type cells to the mutant, or going through an intermediate step, which requires a duplication event, but then having a fast mutation rate? And we studied this with this sort of agent-based model. Here is an agent-based model that you show the expansion of the 2D colony, and you see these various types of mutants popping up. And here are sort of the growth laws that you can see. So you start off with the wild type. You can either go directly, get your mutation, or you can get the duplication or amplification first, and then the mutation. And these are the different types that correspond to up here. The wild type grows here. The mutant that has been generated directly is this yellow line. The mutant that has been generated indirectly through the amplification is this line. And you can see that at early times, when the cell numbers are low, the amplification pathway really doesn't matter. It's much less important. But as the population grows over time, the mutants generated through this amplification pathway does become the dominant mode. And this is in a 2D spatial simulation. And this is exactly done by running an agent-based model and then using the scaling laws to extrapolate to large sizes, which are computationally not feasible.
These are all neutral mutants?
Sorry?
These are all neutral mutants?
This is, I believe, maybe neutral mutants. Yeah, I need to look. There may be slightly advantageous. I need to look up. I need to go back to that, but it shouldn't matter too much. So here you get a summary of these results.
It says S=0.01, so it's slightly advantageous.
Oh, yeah, it's slightly advantageous. Yes, exactly, yes. So what you have here is a measure that is the relative contribution of the duplicated cell pathway relative to the direct pathway. And when this measure hits 1 and goes beyond 1, then the duplication pathway is more important. And so we look at this measure as a function of population size, and we look at it in 2-dimensional space, in 3-dimensional space, and exponentially. You can see that for a 2D growing cell population, it becomes the amplification pathway becomes important quite quickly. For a 3D spatial model, it still reaches it within reasonable populations. This is 10 to the 12, but it's a bigger population than you get. If you have an exponentially growing cell population, the amplification pathway will never be important. And so this is a nice demonstration that shows how space can really be important in determining what is a driver of evolution and what is not. And this gene amplification pathway, similar things apply to things like genetic instability. You can go develop mismatch repair deficiency, then have a higher mutation rate, same thing. So there are interesting things going on here. And what this analysis is telling us is that the spatial structure is really important. So okay, to finish up, I want to switch gears a little bit and talk about an interesting thing we found, and we're going to go away from expanding population, but look at constant populations now. Why are constant populations important? Healthy tissue obviously is at homeostasis, and as I mentioned, in multi-step carcinogenesis, you have periods of stasis where not much is going on. So when you have a constant population, the fixation probability of a mutant is an interesting evolutionary measure that we often consider. And so this is explained here. You start off with a wild-type population at equilibrium. Then you introduce one mutant cell into that wild-type population. And then 2 things can happen. Either the mutant cell population grows to fixation, takes over, or the mutant population goes extinct and it's gone. And we ask, what is the probability that the mutant is fixed? And that has been done by many people in many different situations. And in mixed system, for example, the Moran process is an important tool to study this. And things have been calculated. It's pretty well understood. What is the fixation probability of neutral mutant is 1 over n, where n is the population size. If you have an advantageous or disadvantageous mutant, you can calculate the fixation probability of those mutants. And typically what people assume is that there is a change in just one parameter, for example, in the birth rate or in the death rate of the mutants, and then things are very well understood. Now, I want to concentrate on a particular type of mutant that is found in all sorts of walks of life, if you look at it. If you look at an organism that has a higher birth rate, it often comes with a higher death rate. This happens in the field, in animals, but it also happens in tumors. Since we're talking about cancer here, I'm showing some data from non-Hodgkin's lymphoma. This is the rate of cell death, and here's the rate of cell division, essentially mitotic index versus apoptotic index. And you can see a clear correlation going on. So there are certain mutants that are around that have both a higher birth rate and a higher death rate. And there's an interesting concept that has been studied in the literature. And this is a paper by Josh Plotkin's lab. And they studied what is called quasi-neutral mutants. And a quasi-neutral mutant is defined by having a higher birth rate and a higher death rate. The mutant and the birth rate and the death rate are multiplied by the same constant factor, which we call tau here, which is a turnover factor. So we can say that the mutant can have a higher turnover. It can have a 10 times higher birth rate. It can also, and it simultaneously has a 10 times higher death rate. And the interesting part about those mutants is that they really have the same fitness, the same number of offspring that they leave. The lifetime reproductive success of such a mutant is identical. You can see that here. If your birth rate is 0.1, death rate is 0.01, your average number of reproductive output is 10. If you increase both by 10, it's still 10, right? Same fitness, or same reproductive output, I should say. So you can study this by running stochastic processes of this kind of Lotka-Volterra competition, ODE, and then you can look at the fixation probability of those mutants. And remember that for a neutral mutant, the fixation probability is 1 over n. What Plotkin et al. Found was that it is not 1 over n, but 1 over n times a corrective factor, where the turnover is in the denominator here. So it's lower fixation probability than neutral. So it's interesting because the quasi-neutral mutant has the same reproductive output as the wild type, but its fixation probability is lower than neutral. Is it disadvantageous? No, it is not. to see that, look at this property. We have not the fixation probability, but the fixation probability times the population size. If you have a neutral mutant, that n times 1 over n is constant. For the quasi-neutral, it's lower, but it's also constant. For a disadvantageous mutant, this drops exponentially, right? So the quasi-neutral mutant has a lower fixation probability than the neutral mutant, but it does not act as a disadvantageous mutant. Why does it have a lower fixation probability compared to a neutral mutant? This is to do with stochastic dynamics. If you have a higher turnover, you simply have, in the same time frame, more division and death events. Since you have more death events, it is more likely that the mutant is going to go extinct, and that's why you have this phenomenon. So we took off from there, and we started asking the question, how do these quasi-neutral mutants behave in a spatial system? So we started off with a patch model, as I mentioned. In each patch, you have basically stochastic dynamics according to Lotka-Volterra competition equations. You have movement around those different patches. And then we determined the fixation probability of the mutants. And there's something very interesting that is occurring here. So these are the fixation probabilities themselves. This is a non-fragmented system, like a perfectly mixed system. This is the fragmented patch model. And so your fixation probabilities go down. Now let's look at the total population size times the fixation probability, which for a quasi-neutral mutant was constant, similar to a neutral mutant. And here you've got your non-fragmented population is constant, but in the patch model, it goes down like a disadvantageous mutant. So now you have the really interesting observation that you have a mutant that has the same lifetime reproductive success as the wild type, but it turns over faster because you multiply both birth rate and death rate with tau. Although it has identical reproductive success, it acts as a disadvantageous mutant, a truly disadvantageous mutant, and will be selected against. And so this was a very interesting insight, and you can do some calculations with that. You can take a coarse-grained approach where you assume that a patch, a deme, is either mutant or wild type, and this transition happens very fast. Then you have probabilities of conversion from one type to another, and you can define your mutant fitness, and you can figure things out. One of the important things that comes out of this is that this requires populations to be small within the demes. If the populations are large within the demes, then this effect disappears. So okay, so that's quasi-neutral mutant. The next thing we did was something much more biologically realistic. A quasi-neutral mutant is something artificial. You multiply both birth rate and death rate by the same constant. This never happens in biology. So let's assume that we have a mutant where you multiply birth and death rate by this constant, but the birth rate also is higher for the mutant. So you have a selection here, 1 plus s. So this would be an advantageous mutant, according to all thinking so far. So certainly if this turnover parameter tau is 1, then you have an advantageous mutant going on here. And if it's less than 1, you have a disadvantageous mutant going on. So what happens if you change your parameter tau such that it deviates from 1?
Is it S greater or less than 0? S greater or less than 0?
Yeah.
0, yes,
Yeah.
sure,
Yeah,
sorry,
yeah.
yes, absolutely.
You
Yeah,
vary
sorry.
application.
Yeah, well spotted. Yes, 0. So let's see what happens here. So you start off here with a mutant that has a higher reproductive output. So it leaves more offspring than the wild type. So by all means, it should be advantageous. If your tau is equal to 1, then it is advantageous as expected. Your fixation probability is essentially S, which is 0.01. Now you increase the turnover of the mutant. It still has a higher lifetime reproductive output compared to the wild type, but it turns over faster. What happens, the more you turn over, the more this mutant fitness fixation probability drops, and it actually becomes less than neutral, and it turns into a disadvantageous mutant. And here you have a case where a mutant is selected against, although it has a higher reproductive success than the wild type, which is yes.
Is this where you inject one mutant into the population?
Yes.
If you inject more, this effect goes away in some sense.
Right.
Hang on.
Okay.
Yeah, wait.
Okay.
We can come to that in the next slide.
Okay. Yeah, yes.
Yeah. So here is the same for disadvantageous mutant. 1% disadvantage if your tau is equal to 1, so usual case. It's disadvantageous, highly disadvantageous. Now we reduce the turnover so that they divide and die less, but they still leave less offspring in their lifetime. Your fixation probability goes up and eventually becomes advantageous. So you have a mutant that leaves fewer reproductive offspring, but is advantageous. So let's come to the, yes?
When you say fixation probability, is this in the context of a Markov model, or is this somehow a deterministic model?
No. This is a stochastic model.
So it's like a Wright-Fisher type of model.
Wright-Fisher is a constant population. We use a Gillespie of the logistic Lotka-Volterra thing, competition model. Yeah.
So what is Darwinian fitness here? Is it fixation probability, or because you write this thing that kind of the lifetime reproductive success and Darwinian fitness?
Okay, let's not use the F word then. Fitness. I think of fixation probability if it's selected against, right? If it has a lower than neutral fixation probability, then it acts like it's selected against.
Right. Okay.
So that's clear, I think, than talking about fitness, which becomes more murky.
You should never do that.
Yeah. Okay, so the question just came up, just to finish up, what if you don't just place a single mutant into a population of wild-type cells? Here we have now a process where you start with wild-type. This is the Gillespie simulation of these Lotka-Volterra systems that I showed you. And you have a mutation rate where the wild-type constantly gives rise to mutant at a defined mutation rate. And here we have I believe this is a mutant with a lower lifetime reproductive output than the wild type, and we start with tau equals 1, so same turnover, and here's exactly what you expect to see. The wild type wins, and the mutant is present at what you call a selection mutation balance. It's selected against, but it's produced by the wild type, so it kind of fluctuates at low levels. Now we will lower tau, and what happens is that the mutant with a lower lifetime reproductive output acts like an advantageous mutant and sweeps in. Now you can do the same thing opposite. This is an advantageous mutant, or a mutant with a higher lifetime reproductive output. Tau equals 1. What happens? It sweeps in, as expected. Now you increase the turnover, still has a higher lifetime reproductive output. It persists at a selection mutation balance. So that underscores exactly that point, that it's not obvious anymore how lifetime reproductive success correlates with selection if both birth and death rates are increased in those mutants. And this is with the deme structured model. You can do the same thing with the spatial model. I don't want to take much more of your time. This is just the agent-based model, and you can do the same simulation. You have the disadvantageous mutant with tau equals 1, selection mutation balance. Here the green is the mutant. And if you reduce tau, still has a reduced lifetime reproductive success, the mutant moves in and sweeps in. On the other hand, if you have an advantageous or higher lifetime reproductive mutant, tau equals 1, you get the mutant sweeping in green. If you increase your tau, it persists at a selection mutation balance. So again, the equivalence between an agent-based model and a patch model, which is sort of a theme in all of these things. So with that, I want to kind of wrap up. What is important, the spatial structure can really have important consequences on evolutionary dynamics. And this can be found in relatively simple models of spatial dynamics that I explored here. All of this can form a basis, which still applies when you look at tumor cells that have all sorts of other complexities, cell-cell interactions, microenvironmental interactions, and so on. But this is sort of very interesting, very basic stuff that should carry forward and that should inform our thinking of cancer evolution, because cancer is an inherently spatial process. And with that, I want to have acknowledgements. Current lab at UCSD, former lab members and collaborators who contributed to some of the results that I was talking about.